you a little bit about the work that we've been doing in machine learning and its applications in biomedical research. I am a computer scientist, so my area of interest, I am happy when I'm developing algorithms. And I was quite terrified of biology in my school days, but then it so happened that a student joined me who got me interested in this area. It was a two-way traffic. He taught me biology, I taught him computer science. So it was very convenient in that sense. So I learned a little bit of biology so that I understand what I am doing or the application that we are doing. So artificial intelligence these days, everybody is hearing about artificial intelligence. It's going into domains never thought of before. What is artificial intelligence, how it stands vis-a-vis the machine learning that we also talk of, and that is in the title of my talk. And in these days, we cannot escape this term deep learning. It's everywhere. So artificial intelligence is the superset. A part of it is machine learning. There are things which are in artificial intelligence which is not machine learning. And a subset of machine learning is deep learning. Machine learning essentially means the computer, the system, will learn from the data that is given to it. Artificial intelligence could be like expert systems, which is not machine learning. Rules are coded there, and then the system, it reasons it out through those rules which are already put there in the system. Those are expert systems. But machine learning is, I give it sufficient amount of data set, and it tries to learn things which without being told explicitly. So, as I said, AI, we try to program machines to mimic human behavior, and now we are in this era of deep learning and generative AI, where even in, when we are doing, let's say, neural networks, artificial neural networks, traditional ones, we have to tell the system that these are my inputs and this is the expected output, and then tell me. I have to extract the features. I'll come to it in one of the slides, it's there. Deep learning has removed that requirement also. You just give it raw data and it tries to learn the input-output function. So I'll not spend time because time is very limited here. Turing test is something which was devised in the 1950s to define when can we tell that a system is artificial intelligence. Turing test passed to a certain extent. CAPTCHA that we do every day is a reverse Turing test actually. So AI, as I said, AI is these days everywhere. You think of city planning, you think of autonomous vehicles, healthcare, agriculture, industrial automation. It's IoT, it's Internet of Things. You have many devices, many sensors taking input all the time, and then you reason it out and you analyze that data through artificial intelligence methods and come up with some predictions or summarization, etc. So machine learning is when you are given, for example, pictures of cats and dogs, and you are given a new picture now. You did not see it before. You have to reason it out that this is a cat. Now to come to healthcare in particular, AI and ML in healthcare, various aspects of healthcare, we can find applications of artificial intelligence and machine learning. For example, you have the patient management systems, the electronic health records. There, you can use artificial intelligence methods to make various types of predictions, looking at all the data which is there. You can improve the diagnosis, improve diagnostics, better patient care, personalization, survival prediction. Also, using AI and ML methods, we try to bring down the costs as well, improve population health in general. So various aspects are affected by AI and ML. And an MIT report, a part of it is I've taken from an MIT report. It says there have been different revolutions in healthcare. The first one, of course, it started with the discovery of the structure of the DNA. Then looking at entire genomes, looking at what makes up the genome, understanding the genetic basis of diseases, looking at data in multiple modes from different sources of data. We try to analyze and come up with our analytics, come up with predictions, looking at genomic, proteomic, expression, images, clinical data, a lot of it. And now we are in this convergence phase where all the disciplines are coming together to solve the problems. We have the life sciences, physical sciences, at the core in engineering, technology, mathematics, statistics, everything has to come together now if we are to really solve the problems that is facing us, facing humanity. And not just in healthcare, I guess this is true for any problem, climate change, environment, any problem that you can think of, it's an era of convergence now. So to just put it in perspective, we have the cell, inside the cell, the nucleus, we have the chromosome, and if we look at it in detail, it is the double helix here, and if just one quick slide of the central dogma of molecular biology, where we know that parts of the DNA is the gene through the process of transcription, we have the messenger RNA because it is carrying the message for the corresponding protein, and then from the messenger RNA we have the peptide, which is a sequence of amino acids. Without going into the details of all these, so we know that we've already heard from the previous speaker about the proteins and how it folds, and then we finally land up because of various interactions with the tertiary structure of the proteins, and proteins are sort of omnipresent, doing variety of jobs in almost anything that we can think of. So what I'm trying to now do is to give you a flavor of different things, four things that we've done in our laboratory from, let's say, about 10 years back to what we are currently doing, because it would be very difficult to explain each in detail in these few minutes that we have, but just to give you an idea of what's going on, that this is in my group. So I started off in computational biology with this particular problem of looking at small RNA molecules called the microRNA. And this was when microRNAs, not a lot of research had been done in microRNA, so we were looking at that molecule, of course, computationally, all computational algorithms and the data that was already publicly available. We did a fairly comprehensive study of the involvement of microRNAs in various diseases, in particular in different types of cancers, and found that it was very heavily implicated in almost all types of cancers. So without going into the details of the biogenesis of microRNAs, what happens is a normal protein coding gene would, inside the nucleus, form the mRNA, the mature, it would come out. The mRNA would normally have produced a protein, but at some point this microRNA gene, which is also in that same genome, gets active and produces a small RNA molecule called the microRNA. It's just about 20 to 22 nucleotides long. It's the mature microRNA. What it does is it goes and sits with this mRNA in the region preceding the actual in the UTR, it is called, of the messenger RNA, of its target messenger RNA. It either goes and represses the translation of this messenger RNA into its protein, or it simply degrades this messenger RNA. The effect is the same, that the protein which was originally getting produced does not get produced anymore. So this is how microRNAs typically work. And we were studying the direct effects of microRNAs. We were also studying the timed effects, meaning secondary effects of microRNAs. So we had a fair amount of publications in that area. But what I will just tell you about is a machine learning problem that we tried to solve there, which was a classification problem. So we wanted to know that if a new microRNA comes up, what is it doing there in the system? In order to understand that, we have to figure out what messenger RNA it is targeting. If we are able to figure that out, then we would know that this messenger RNA was producing this protein. So that would mean that this particular microRNA has some effect on that protein. So it gives us some handle on that new microRNA that we've discovered possibly. And it was posed as a computational problem of classification, very well known. There are many well-known classification techniques. So the problem essentially was, I have a set of pairs, I have many pairs, microRNA and mRNA pairs, which are known to be targets or known to be non-targets. So people, for example, for this pair, biologists have actually experimented, put them together and found that this microRNA has no effect on the messenger RNA. The expression of the messenger RNA does not change. So that means it is a non-target, very likely to be a non-target. So this was the problem. We had a set of targets and a set of non-targets. These are microRNA and mRNA pairs. Now given a new pair about which we do not know whether it will target or it will not target, we have to infer, we have to predict whether this is a possible target of this microRNA. So this is basically a classification problem in computer science terms. The problem here, there were some prediction methods which are already available, but when we checked out all these algorithms which are there at that time, the results were widely varying. Okay, and we figured out the machine learning methods, typically they need training data. And training data means there should be enough positive data, there should be enough negative data in order to train a classifier. The problem is, in this scenario, there was a lot of biologically validated positive data, positive meaning targets. A lot of it was available, but very few non-targets were available because biologists, when the experiment failed, a particular microRNA did not target a particular messenger RNA, they did not report it. So there was a lack of negative data, and therefore the machine learning people, what they were doing is they were artificially generating the negative data. And in that process, that artificially generated negative data became very much different from the true negative data. It was also quite different from the true negative data, positive data. So what happens is, if you have a positive class here and a negative class here, which is very distinct, it is very easy to classify, put a boundary in between. But then the real data is not that distinct, it is actually very much overlapping. The negative data has a lot of positive, has a lot of characteristics like the positive data has a lot of characteristics like the negative data, so real data is not that simple. So since people were actually working with artificially generated negative data, which made it quite different from the real negative, so there was a wide variation when it actually was applied to real data sets. So what we did was, we came up with a sequence of steps of generating the negative data. We actually started from a set of predicted positives, predicted targets, and from there we filtered off, using very, very stringent methods, anything which had a possibility of being a true target. Anything which had a possibility of being a true target was eliminated from that set of predicted positives. And now what we were left with after various steps of filtering was a set of negative data which was quite, which had positive characteristics, because these have come from predicted positives. But it had very, very less chance of being a true positive. And indeed, the classification was standard. The results are here. You see, this has a very high, it has a high true positive rate and a low false positive rate. So this is the quadrant where we would like our algorithm to be in. This and another very popularly used method, target scan, it appeared there. The others were either too conservative or too, too optimistic. So they were performing at different levels. And we used this method. In addition to that, we actually, we made a regulatory network inside the cell. So gene and transcription factor networks are well known. Transcription factors switch on or switch off the different genes, and transcription factors themselves, being proteins, would be generated from some gene. So there's a gene regulatory network in action inside the cell. We at that time put in also the microRNAs. That was sort of the first work where microRNA induced transcription factor gene regulatory network was studied. And we studied it for breast cancer and colon cancer, did some graph theoretic analysis, and came up with some interesting results where certain microRNAs, which are not known to be involved in those cancer types, they those were got predicted. So that was interesting, because this was, these two were new microRNA predictions for breast cancer, and this microRNA was a new prediction for colorectal cancer. Our work had to stop thereafter because we could not take it further, but we published, hoping that somebody else somewhere would pick it up. And people did pick it up, validated for the one microRNA got validated for breast cancer. I have not checked out later what happened to these others. Quickly again, because I don't have time, we also, with one of my students who did her PhD, we looked at the drug design problem, posed it as a problem of multi-objective optimization. So what we know, for example, this is a flu drug. This flu virus. So this virus has the RNA inside. It has several proteins which helps it bind to the surface proteins of the human cell, helps it multiply, helps it again escape from the human cell and go and infect other human cells. So the point is, can we target any of these viral proteins which is necessary for the survival of the virus through small molecules which could be drug-like molecules. So this is again, I don't have time to go into this, but a team from Australia, led by Professor Peter Coleman, actually designed a small drug molecule using such methods, which is rational drug design, methods of rational drug design. And this small drug molecule called Relenza, it was actually discovered through these principles. We also tackled this problem, but we were looking at the problem from a multi-objective optimization point of view, as I said, and we were looking at different objectives which we want to optimize. For example, the molecule themselves should have very low energy component, it should have high oral bioavailability, etc., etc. So different objectives we were trying to optimize at the same time. And we were looking at multi-objective optimization techniques that we had developed in our own laboratory. So it is not that you have 5 different objectives, you just combine them W101, W202 and come to one objective and then you optimize. That is not how this will work. It is all the objectives are kept separate and optimized simultaneously. So what you get is a set of solutions at the end. So what we were doing is we were looking at these 3 objectives which we wanted to optimize, and we were targeting the REC-A protein of Mycobacterium tuberculosis. REC-A protein, as there was a discussion in the morning, it repairs the bacterial DNA, and therefore the antibiotics which harm the bacterial DNA, REC-A is responsible for that DNA repair. So REC-A is quite a well-used target for different viruses, including for bacteria. This we were using for bacteria, Mycobacterium tuberculosis. And what we got, interestingly, was several inhibitors. Our method gave several inhibitors which are already known for REC-A target, giving us confidence in the method that we developed. At the same time, several other inhibitors got proposed by the system that we developed, and we did some QSAR studies, quantitative structure activity studies for these molecules also, trying to show that these could be potentially good molecules. But thereafter our expertise ended and we were unable to proceed further with our limited expertise. Now we published, and then hoping again that the methods will be taken over by other researchers who can actually look at the biology part of it and test it out. We were also doing some single cell, so clustering methods. We have where very small rare cluster types, rare cell types could be detected. And I don't have time at all, I can see the clock ticking. So we had, for example, this data type where very small clusters also could be detected. Ordinary clustering methods would be unable to detect such very small clusters. And in cancer in particular, with such a heterogeneity in the tumor cells, it is very important to detect rare cell types. This method was able to do it. And finally, we are now working in these areas. This is a current area. As I said, from raw data, you do machine learning like this. So you have to extract features, do feature engineering. Deep learning has done away with that feature engineering part, but with graphs, if you are working with graphs, then even this is machine learning, application of machine learning is also very difficult. So what we are doing now is working with something called graph neural networks, where given the graph, we are coming up with embedding of the nodes. Embedding in such a way, these embeddings would be learned in such a way so that nodes which are similar to each other, their embeddings would also be close to each other. So that is, so now for every node you would have a set, so for every node you have a vector of numbers, but now you have a data set of numbers now, apply whatever machine learning technique that you are fond of. So this is the good thing, we are working with a network of drugs and proteins and genes and trying to solve problems of adverse drug-even prediction, drug-to-drug interactions, and if there are adverse effects, we are trying to predict those. I'll stop with that. There are some references here, old references as well as the new ones here. And thank you very much for your patience, for listening to me, my acknowledgements to my institute and all other agencies, and thank you very much.
With some trepidation, where is it? With some trepidation, I'm trying to pose a few questions. I'm afraid we'll run out of time, I know that. And there's a lot of material, very interesting material to be examined. I take up the first few sets of slides wherein you draw a Venn diagram for that machine, artificial intelligence, machine learning and deep learning, that's one. And there is a reference somewhere in between to the double helix and things of that sort. And finally, you have a graph network and all that. For the concerning these three, I have three questions. I'm sure they will get impatient with me. I apologize to them in advance. With reference to the first one, let me consider machine learning first. You have a set of photographs, chimpanzee, human face, and all that stuff. Then the question is, you are going to train the network, whatever it is, based upon these things. But then the problem is, what is the amount of data required to be able to generate meaningful parameters of your network? That is one. Thirdly, with reference to deep learning, what exactly will you achieve without tutoring the machine? Because deep learning is a self-learning, some sort of self-organization you can call. I mean, if you want to treat it that way. No, that's why I know we can discuss it at length. So what type of mechanism is involved in deep learning which will facilitate discrimination automatically? So that is one set of questions. The second one is on the... No, because I'm sure they'll be impatient. No. And the second question is with reference to the double helix and all that. Linus Pauling and others have been fascinated, almost hit upon that idea, didn't succeed. Assume that we have all the data required until Linus Pauling's time, and somebody else, Crick and Watson, they came out with the result, and then Nobel Prize and all that stuff. Now suppose I want to come out with a machine which can declare, hey, there's a double helix based upon the data. What type of processing will you need to do that? That is AI. Suppose the AI, you say, is going to solve all our human problems with that type of data, with no other knowledge. I'm very strict. That means there's the boundary. Can AI declare, hey, I have got the double helix. I will get you a triple helix for some other data. That's it. The third one, on the graph network. What you are trying to do, if I understand your graph explanation, you are trying to see whether there is a clustering mechanism embedded in the clustering in the data, which will facilitate your node choice. Thank you for your patience. I apologize for all the questions.
So first question, it's not all self-organizing and self-training. For example, in deep networks, you would have to give it enough examples of, let's say 10 different things, 10 different types of images, meaning images of buildings, images of clouds, images of glasses, images of snow. You have to give it enough number of examples and tell it that this is the image of so-and-so, this is the image. So you have to have the tutoring there but only the input output.
Sorry, I have to interrupt in this context. In early
Then I
literature,
think we have to take it up.
no. One point here. In early literature, there was a terrifying comment. What was that? You are over teaching. You are saturating. Even a minor departure will not be accepted. So over teaching is also very, very unsatisfactory. You have to allow so that the machine can do some
Now,
good
actually,
job.
the point is we have to learn millions and billions of parameters. So data sets have to be really large to train the deep learning models. The small data set, this is not going to work. So yeah, and the other questions, I think we would need a little discussion. I've forgotten also many of the questions. But we will discuss, certainly.
In the 90s of the last century, people looked at combinatorial chemistry as a panacea for drug discovery. They discovered that they made millions of molecules, but the whole concept miserably failed. They didn't come up with a single drug molecule. This machine learning and AI and all that, I'm just worried. What is
No,
it?
you don't need to be worried. You see, it's at least at this point of time, we are not seeing machines replacing humans.
No, I'm
So
not
that
worried.
human expertise will
I'm
be there.
already retired, so I'm not worried.
But you said you were worried. So, but the point
yeah,
is,
okay.
you see, you cannot do without these techniques anymore. You just cannot do. At this point, still, it is still, we are developing the algorithms, we are controlling it, and so on and so forth. But where things are going in the artificial intelligence research, the entire world is learning. That, in fact, there are calls for stopping AI research, putting regulations in place first, and then continue.
I'm supportive about that. See, I'm from IIT. And I can see that all the engineers are doing all these calculations without taking into consideration the opinions of chemists in the back.
No, that's wrong.
So that's,
And that is
and
something.
they are talking about drug discovery at the same
I
time.
understand absolutely. And what we have found is, see, it has been easier to collaborate outside India.
It is
than in
easier to say
India.
no
Sorry,
to
but
the
this is a fact.
gentleman.
And that level of respect for each other's expertise is sort of missing. A biologist will think, I know everything, and there are software which are out there. I just take out of the shelf, and I'll apply and do it. You see, if real problems are to be solved in a meaningful way, experts from all these different domains have to sit together and solve the problems. In India, that has been a real challenge. We've tried, at least thrice I've tried with my students, ended up with acknowledgements in the papers. So naturally you can understand that PhD students are also getting demoralized.